finalizing these lectures, we did not have the calendar yet. And sometimes in years past, these lectures haven't been back to back. So we're actually going to be able to skip the first, well, not to the next slide, but several after that because they're reviewed, just, in case these lectures were not back to back. But before we hit this, let's take a side step and talk about some terminology because I'm going to use some terms today. There'll be some designations on the slide that may seem a little wonky. All right, so terminology. So when we talk about a CD4 T cell, right, it's expressing CD3, right? All T cells, at least all conventional T cells express CD3. And it's expressing CD4, right? We're all in agreement on that? So one way that you can write this is CD3 plus CD4 plus. That means it's positive for CD3, it's positive for CD4. Just a shorthand, we typically don't do that because we're educated immunologists and we know that it's there. And we can even say just CD4 because we know by default, if it's CD4, it has to be CD3. Okay? All right. But how do we designate the absence of a marker? Because a CD4 T cell, does it express CD8? No. So we could, in extreme longhand, go CD3 positive, CD4 positive, CD8 negative. Because, as really well-educated immunologists, we know a T cell may express CD8. This one just doesn't. All right. Everybody with me? All right. Why am I making a point of that? Is because once we get to this lecture a little bit, I'm going to say double positive and double negative. That's part of the T cell. This is going to be a very active process. Sometimes those developing T cells are not going to express anything but CD3. They're going to be double negative. So that's CD3 positive, 4 negative, 8 negative. We put the 4 negative and 8 negative just to say, yes, we know that it has the potential to express that, but it's not at this point in its life cycle. Flip side, double positive, it's expressing both 4 and 8, and that's how we would write it. All that makes sense? It's really simple when you think about it, but you'll see this all the time because when you're talking about genetic issues, genetic diseases, or if you're talking about blood, leukemia, lymphomas, and you're going to see the expression of a protein that's not normally expressed by that cell, or the loss of a protein. And so that's why you may see your lymphoma as a CD34 positive, because that mature B cell should not be expressing CD34, but it is because it's cancerous. Okay? So it's not just in T cell development that you may see those pluses and minuses. All right? Okay. All right. So, yep, these are the slides that were reminders if this lecture might have been next week, which has happened. All right, so we're going to focus on lymphocyte development. So we've got 3 major steps, and there's actually going to be repeats of these steps. There's going to be a proliferation step, because this is all about populating the periphery, making enough cells. We're going to have expression of those antigen receptors. Now that we know how to make them, we need to start making them. And then there's going to be testing of those receptors. Did we make a functional receptor? Does it have the right sequence? Does it have a constant change so that it can signal? And does it react with self? All right? And then based on that pattern, we're going to test it. If it passes the test, that's going to be called positive selection. If it fails the test, a variety of reasons it can fail that we'll talk about, that's going to be called negative selection. This is a huge thing that confuses people. A lot of the images, just because you can't draw them both, but a lot of the images will say either positive selection or negative selection. It's not just one. Think about this as flipping a coin or as walking down a road and you hit a fork in the road. That developing cell must make a decision or must pass a test. Do you take a test and you're guaranteed to pass it? We wish. But no, there's always that option. Not that you all have that, but there's always that option. It's a pass or a fail. It's not just that one road. All right, and so when we get to these checkpoints, That's when the developing lymphocyte is tested. Always remember that it can go either way, all right? The reason the images only put one is because it's which step is most important to determine, which result is most important at that point in development. And hopefully that'll make a little more sense here in a bit. All right. Okay, so we do this so we can generate lots of specificities, the diversity of the immune response that is not specific per cell. All right. This starts out in your primary lymphoid organs, the bone marrow and the thymus. T cells complete all of it in the primary lymphoid organs. B cells finish it out in a secondary. And then we have those different checkpoints through the maturation process just to make sure that the T cell receptor or the antigen receptor is functional and that we can eliminate anything that is self-reactive. All right, and talked about this before. We've got those repeated cycles. There's proliferation. There's a checkpoint. If it passes the checkpoint, it then goes into the next developmental change. If it fails the checkpoint, here's the path in the road. Passing the test, failing the test. If it fails the test, it's going to die by process of apoptosis, process of neglect. Again, those processes of self-destruction, cell suicide. All right, so how are we going to start? Both the T cells and the B cells come from a common progenitor. So first, there's the hematopoietic stem cell that's in the bone marrow. And I was so busy answering questions, I didn't switch over the pointer, so I get to wave my arms again. All right, so you have the hematopoietic stem cell. It gets some sort of signal that says, okay, I'm going to become a lymphocyte. All right, then that hematopoietic stem cell will differentiate into a common lymphoid precursor, the CLP. All right. Then it starts interacting with the stromal cells in the bone marrow. So all the cells lining the interior of that bone. Signals from those stromal cells will then differentiate that common lymphoid precursor into either saying, I'm going to become a T cell or I'm going to become a B cell. And then you get those very earliest, but they're committed to that pathway at that point. So they're pro-T cells or pro-B cells. All right? Everybody with me? All right. So then the T cell says, all right, I'm going to be a T cell. I need to get going. It becomes a double negative cell, leaves the bone marrow, and goes to the thymus and finishes the process. All right? The pro-B cell stays in the bone marrow, continues its differentiation through different steps, and you can see that down here at the bottom, interacting here at the very bottom, and it's kind of gray, with those stromal B cells, those stromal cells in the bone marrow. Through those different interactions, once it starts expressing IgM, it's going to leave the bone marrow. It still is immature. It's going to leave the bone marrow as a transitional B cell, and it's going to start expressing IgD. So there's that first isotype switch. At this point, it's going to express both IgM and IgD on its cell surface. At that point, it's a mature, naive, but mature B cell. All right? IL-7, during these early stages, is the growth factor. So IL-7 is being produced by the stromal cells in the thymus and in the bone marrow. And that's your first growth factor for your T cell and your B cell. Okay. That's from the satellite view. That's today's lecture. All right. Let's get a little more specific. Actually, you just talked through all of that. So, just got a little too excited. This shows the IgD and the IgM, everything happening in the bone marrow, and then on its way, or once it gets to that secondary lymphoid organ, the spleen of the lymph node, that's when when the B cell becomes a mature B cell with a mature receptor and can be activated. Okay? All right. This is a very active process. This, by no way, shape, or form would I ever expect you to know 90% of what's on here. But I want you to understand the dynamics of this. So here are T cells here are B cells. If you go from left to right, that's its developmental process. It's guided by the differential expression of different receptors, as you can see here in the pink. So at different points in the developmental process, some receptors are expressed and then turned off, while others aren't expressed and then they're turned on. All right? So this is the maturation process. Right? This is how we are able to type leukemias and lymphomas, because a lot of those blood cancers start expressing developmental antigens and developmental receptors. Right? So that's why I want to kind of give you a glimpse into what happens here. The things that you might want to know: you've got your IL-7 receptor. They've got to express that receptor because they've got to be able to respond to IL-7. Remember, that's the first growth factor for that proliferation event. Then, if you're a T cell, you need to be able to express CD25 later in the process so that you can convert from proliferating in response to IL-7 to start proliferating in response to IL-2. Because once you get out into the periphery, that is your proliferation factor, only IL-2. All right? And then we've got some of the lineage-specific markers. CD3 that says, hey, I'm a T cell. B220, CD45 that says, hey, I'm a B cell. Okay? All right. Let's look in fine detail about what's going on. So now we're going to talk about T cells. Here's a cross-section of the thymus that hopefully looks familiar from your organs lectures with Dr. Prater. So you've got the medulla and then the cortex here on the outside. Those open spaces are vessels, whether they're lymphatic, whether they're blood vessels. Formal pathological name for them, the high, or histological, high endothelial venules. And that's where the cells both enter and exit the thymus, okay? So if you look down at the cartoon, they come into the thymus through these HEVs, go through the medulla, and hit the cortex, right? This really dense region, that's the cortical-medullary junction, all right? So that's sort of the transitional state. They go into the cortex, go through some of the differentiation processes we'll talk about, circle back around, finish, have their final exams here at the cortical-medullary junction or in the medulla, and then exit through the high endothelial HEVs again. All right? Okay. So, the important factors here, those thymic epithelial cells, the TECs, whether they're in the cortex, so the CTEC, or in the medulla, the MTECs, they are the source of antigens, and they are the source of a lot of the cytokines that are supporting this process. All right? Then you have your soluble factors, the cytokines and the chemokines, something that has to direct the traffic, something that has to induce the proliferation. And then you have those specialized antigen-presenting cells, especially down here in the medulla for those last stages of the developmental process. These APCs are not found anywhere except the thymus. They're specialized subsets of DCs and MACs. All right? Okay. All right. The thymic education process that we've talked about a couple times, or we've referred to, that's your positive and negative selection. We're going to have, for a T cell, 2 exams, 2 points of that differentiation, that checkpoint that occur and must be passed before the cell can exit the thymus and enter the circulation. And we'll talk about the details of those. All right. So here we go through the process. The T cell enters the thymus as a double negative cell. It's not showing the entry. It's entered and it's already gone up to the cortex, okay? So here's your double negative cells up at the top, all right? And then they start interacting with those cortical medullary cells. And by that point, they have made their T cell receptor, all right? So they're double negative, not expressing CD4 or CD8. And then they migrate down and have their first checkpoint, right, their first test to see, is that T cell receptor made. Can it see MHC? All right, with an antigen in it, and can it signal? If that is successful, that means yes, the T cell is made, it can signal, everything is good, and it passes the first checkpoint. All right. Then it will move down to the corticomedullary junction. It will express both CD4 and 8, so it's now double positive. Enter into the medulla, and it has its second test. What about the antigen that it sees? Does it recognize self? So the first test is really mostly about can it see MHC. The second test is really about what antigen it sees. Does it see self-antigen really well? Or does it just see the peptide enough to say, yeah, I see that there's something there, but I'm not going to respond? All right? It's the Goldilocks principle. We want to see the antigen well enough that we know we can generate a signal, we being the T cell receptor. And then we pass that positive selection, that second checkpoint. In nearly all figures, you're going to see this checkpoint written as a negative selection because that is the most important point. We really want to screen out what peptide, what T cell receptors are specific for self-antigens. So the important result here is we want these guys to fail. So it's a negative selection. Because if they recognize that self-peptide with high affinity, they're going, and they get into the periphery, they're going to cause autoimmune disease. So this is where we want to target those cells, have them be negatively selected before they can get out into the periphery. All right? And then at that point, a second event that happens during that second checkpoint is they establish their preference. Do I prefer seeing my antigen on MHC class 1 or on MHC class 2? If I have a better preference, if I see the antigen better when it's on MHC class 1, who recognizes MHC class 1? CD8? Great. Woo-hoo. So I'm going to downregulate my CD4, I'm going to maintain CD8, and now I have become a single positive CD8 T cell. If I see my antigen a little bit better, if my preference is for MHC class 2, I keep my CD4 expression, I turn off my CD8 expression, and I become a single positive CD4 cell. Okay. Then I leave the thymus, exit through those HEVs again, and I am a mature but naive. I haven't seen antigen, so I'm still naive. But I'm a fully functional, fully licensed T cell receptor. Or T cell. Okay. That kind of makes a little bit of sense. All right. So here's another image that can make it maybe a little more obvious. The exact same process we just talked about. You know, here, last slide, it really gave you a good context for the trafficking. This gives you a good context, in my opinion, for the different events that occur once you're in there. So way off screen over there are the HEVs. Here's here's an HEV, here's the cortex, where it enters, double negative. The first positive selection, by default, flip side of the coin, other fork in the road, negative selection. But if the T cell receptor can see that MHC molecule, it passes the test and moves on. All right. So this is your first checkpoint. Have I mentioned NKT cells or gamma delta T cells yet today? No, today, yes. In this hour, no. Here's where they're unique, or why they're not conventional, why they're the unconventional T cells. At this point, the NKT cells and the gamma delta T cells leave the thymus. We don't know why. We are clueless. We don't know what signal mediates that sends them out. But they leave the thymus once they get to that point. And if you remember, it kind of makes sense because they don't recognize their antigen on MHC. NKT cells use CD1. So somehow they get to this point, they become the single positive CD4, CD8. They leave the thymus, and they leave the thymus already about halfway primed. So once they get into the periphery, they react to their antigens extremely rapidly. They're not going to go through all the processes we're going to talk about tomorrow for for T cell activation. Okay? So that's what sets them apart. It's only the conventional T cells, the CD4, CD8s, that then continue this path into the medulla and through to that second checkpoint. All right? And then here is where they become single positive at the same time that they're having that second test for testing for the antigen. All right? Really important point here. There is no foreign antigen in the thymus. Only self-antigens are expressed. All right? So this is why it becomes very important to know what the affinity is for that T cell receptor, for that antigen. Right? And that is governed by this relative avidity or affinity. So here's across time. This is your T cell receptor. I'll switch slides for a little while. This is your T cell receptor expression. So across time. And then the relative avidity of that signal. All right? So if you look in the red line, that's the just right, if we go back to our Goldilocks analogy. That's the T cell where the T cell receptor can see your MHC with the peptide setting in it. And it can bind just enough to say, yes, I see that T cell. Yes, I see there's peptide there. And I can signal so I can go out and do my job. Okay. That's the goal. That's the T cells that are preserved. Yes, ma'am?
So for the apoptosis threshold, would that mean that it exceeds itself too much?
Hold that. I'm getting ready to answer that. You're thinking about 3 sentences ahead of me, but good job. All right. So if that T cell cannot see the MHC, or it can't signal, that's going to be the line in the blue. That's below the threshold. That red dotted line is the minimum threshold. There has to be enough signal generated to get above that red line to say the receptor is formed correctly and functions correctly. All right? Now, so then it's death by neglect is the formal term, but the cell just kind of withers away and dies. It starves. It doesn't get the growth signal. So above the black dotted line, those are T cell receptors who don't just bind kind of loosely, they bind tightly. That's a highly specific binding event. That means that T cell receptor specifically recognizes that antigen. Those are the T cells that if they get out into the periphery, say they recognize, do you all know diabetes, islet cells in the pancreas? That's what, okay. So that means that if that T cell is specific for a protein made by a beta cell in those islets, you're going to have diabetes if that T cell gets out. So we need to be sure we get rid of that T cell. We eliminate it from the population. Above that black line, that's the apoptosis threshold. That is where there's a strong signal that is generated and sends that cell, starts that cascade so that the cell dies. All right? Does that make sense? Okay. All right. The phase 1, the phase 2, the phase 1 is that, if you go back to the last slide, that's the first checkpoint. The phase 2 is the second checkpoint. All right. And then this is the process where just thinking about the changing in the, from double negative to double positive to single positive, this sort of just puts it into a cartoon and then shows you some of those interactions that must occur. Right? Okay. And then this just summarizes, all of the events that occur and then your different checkpoints. So this is on the, going back to the graph I showed you a minute ago, this is when there's not enough recognition, so the bed's too soft, going back to Goldilocks, and then here's the specific recognition down on the bottom, the bed's too hard, there's too much of an interaction, and it is apoptosis, active signaling, and then here is where the binding is just right. Okay, that light attachment, just so we know that it sees everything it's supposed to see and can signal. All right, and then this just puts together your CD4 and CD8 T cells. They have your alpha beta receptor. One of them expresses CD4, one expresses CD8, kind of obvious, and then where they see their antigen. Your Treg cell has an alpha beta receptor, has CD4. We'll talk about FOXP3 a little bit tomorrow maybe, but more next week. And it sees antigen in MHC class 2 because it's typically CD4 positive. And then your non-conventional T cells, your gamma delta T cell has a gamma delta T cell receptor. Immunologists are really creative, right? It's not MHC restricted. We don't really understand how it sees its antigen, but we know that it's not in the context of MHC. Most often they're CD8, and remember, they go really quickly from the thymus and then go to the epithelial barriers and intercalate into those epithelial barriers. And they're there as sentinel cells waiting to respond. And then your NKT cells. All right, we've got a slide on that in just a second. All right, so your T cell development is all driven by your transcription factors. And so you get, whether it's cytokine binding, whether it's interactions with those stromal cells in either the bone marrow or the thymus, you're turning on different transcription factors that drive that differentiation pathway. For Tregs, this is one of the, and maybe only the transcription factor I will ask you to know throughout this course. Tregs express FOXP3. That's one of their primary signature molecules, as we'll talk about, okay? And here is your Treg cell, and so here is the transcription factor that's turned on during its differentiation process. So in the thymus, we make what are called natural T regulatory cells, because they're the cells that like to push the envelope. So Treg cells are the cells that suppress immune responses. They're not activators, they're suppressors. They turn down the volume, they turn things off. And so, they like to push the envelope because the signal that turns on the FOXP3 is generated because they have a very high level of specificity, very high avidity for that self-antigen. They're just right here at the bubble of being negatively selected. Right? That's where I say they push the envelope. They're at the highest threshold possible, but still making it under the threshold to be preserved in the periphery, all right, or in the thymus, all right. So they're expressing that very high level of antigen. That high avidity turns on FOXP3, and then it turns on the rest of the differentiation process that makes them a T regulatory cell. They also express very high levels of CD25, and we're going to talk about them repeatedly when we talk about tolerance, when we talk about some autoimmune diseases, and when we talk about turning down, contracting the T cell response. These are all functions and processes where T regulatory cells come to play. So natural Treg cells are differentiated in the thymus. All right, we'll get to another type of Treg cell tomorrow. All right, the non-conventional T cells. Basically, we've covered all of this. So just if you want a reminder of that, there it is again. All right. I bring up NK cells here for one reason, because I always get the question, well, where do NK cells differentiate? They're not on your picture in the thymus. Well, they're not because they don't go into the thymus. They are not adaptive cells. They are innate cells. An NK cell is different from an NKT cell. All right? So, yes, an NK cell is a lymphocyte. No, it does not go through the thymus. Okay? It doesn't express T cell receptor. It doesn't express CD3. It doesn't see its activating signals in the context of MHC. All right? It doesn't have any of these checkpoints. All right? So it's an innate cell. All right? B cell development. All of this happens in the bone marrow. All right? And you go through the process of rearranging the receptor, expressing your light chain, your heavy chain. We'll talk about those in detail in a second. Until you get to the level of IgM being expressed. At that point, it leaves the bone marrow as an immature B cell, and it starts to express IgG. At that point in the periphery, whether it's in the spleen or a lymph node, as soon as it expresses IgM and IgD, it is now a mature B cell. Still naive, hasn't been activated, but it is mature and ready to function. All right, so what are the checkpoints here for a B cell? I'll talk to this side for a minute. So you have your hematopoietic stem cell that again is off camera, off screen. Your early pro-B cell that came, hematopoietic stem cell, common lymphoid precursor, then the B cell. The B cell begins to rearrange its heavy chain, right? Its heavy chain initially is paired with a placeholder, not the light chain. Its first test is just to test to see if if the heavy chain is made correctly, all right? So that's what that alpha 5, that's a pseudo light chain, that's the placeholder. Not alpha, lambda. That lambda 5, so that's the placeholder. So test checkpoint number 1, is my heavy chain correct? If it is correct, and we don't understand this process. I mean, you can't really pull out the bone marrow and do things. For T cells, how we know so much about the process, you can pull out the thymus, you can do histological sections and really understand the process, isolate the cells, do all the histological staining that we can't do in the bone marrow. So it's a little fuzzy for some of our understanding of some steps. All right, but we know we've got the heavy chain is only at the first checkpoint. If the heavy chain is properly made, if it can signal, then it proceeds to the next point. If heavy chain is not properly made, the cell goes into apoptosis and dies. All right? Everybody with me? Okay. Then, if it passes that first checkpoint, it starts rearranging its light chain. Like I said last hour, most of the time, over 99%, it's kappa chain. Sometimes it's lambda. So it makes that light chain. It goes to the next point, the next checkpoint, has another test. If it passes the test, it goes out, starts making IgM, and then goes out into the periphery and makes IgD. Right? If it fails that checkpoint, it can try making a second light chain. It may go back to the lambda chain, or it may go back to the kappa chain and try and make another kappa. We don't understand what regulates this process. Sometimes the cell doesn't get that second chance. Sometimes it gets one. The most that we've ever been able to see is around 5 or 6. Have no idea the regulation. But bottom line, there's a potential to make multiple light chains or multiple attempts at making a light chain that is functional. At no point is the heavy chain altered. That stays the same from the initial synthesis. Okay. Does all that kind of make a little bit of sense? All right. So that's your B cell differentiation, your B cell maturation, receptor maturation point. All of this occurs in the bone marrow until you hit that IgM. Okay. What happens in this? So this is different. Once a T cell receptor gets into the periphery, that T cell does not change its receptor in any way, shape, or form. All right. B cell can. So we kind of talked a little bit about the whole isotype switching. We won't revisit that. In some text, and I think Dr. Prater uses this, that this initial is a rough draft of the B cell and the receptor because it can change slightly. It never changes its antigenic specificity. It's always going to recognize the same thing, but it can go through some changes that make it recognize it better with a higher affinity or avidity. And so this is called a process of hypermutation. Each time you see that antigen, you start recognizing it better. You make small point mutations in those CDR regions, those hypervariable regions, that then increase the affinity of binding of that antibody molecule, of that B cell receptor. Okay? That's called affinity maturation. All right. You'll revisit this again with Dr. Prater. So just since this is your receptor lecture, I'll at least introduce it a little bit here. And then as the affinity of that B cell receptor increases, then you have more mature forms of the antibody that are produced. All right? Here's a good model that shows this and comes back to the analogy that I like. So this is at day 7, so this is where you're making IgM. Maybe starting to think about making some, whatever your IgG or Ig is. But as that antibody is made, as those B cells encounter antigen, they start making single point mutations in these hypervariable, in these complementarity determining regions. So you have them in your heavy chain, you have them in your light chain. Remember, that's the little picture of the actual antibody binding site. Yes, ma'am?
So does affinity maturation occur during clonal expansion or afterwards, delivery?
Okay. So the question is, does affinity maturation occur during the activation or at the end of the immune response when you enter the memory phase? It's happening in real time. As that B cell is dividing during the primary or the secondary immune response, it's making these changes. Okay, and that's what we see here. So here it is at day 7. Each of these little burgundy lines is a new point mutation. So at day 14, we've got a lot more. Now you're infected a second time. So you've got even more. And here's a third time. Every time that B cell receptor encounters antigen, it can make more of those mutations. Now, will some of those mutations decrease affinity? Yes. But those cells don't survive. They're outcompeted by the B cell receptors that make a better, stronger binding event. And so it's just competition for resources. Those receptors that bind better are going to make contact, they're going to suck up the antigen, so they'll get the survival signals, and then any B cell that has a lower affinity receptor will fade away. Okay, here is the analogy I like to use. And so my youngest nephew is a basketball player, and so he talks, well, he did talk a lot about Steph Curry, and someone just clicked one day. All right, you're Steph Curry. Or, no, you're not Steph Curry, but you aspire to be Steph Curry. So you're out there doing your free throws. The more you practice, i.e., the more you encounter antigen, you're going to get better. Your binding is going to increase. Your three-point percentage is going to increase, right? Will you become Steph Curry? No. So you do not change from a B cell receptor. Will you start shooting soccer balls? No. Your specificity for antigen will not change. You're always going to be shooting the basketball. You're just going to be doing better. That's affinity maturation. Does that help things make sense? Okay. All right. Here's isotype switching. Same thing we showed before. And so, question at the break that maybe can help this a little bit is once you've done your VDJ recombination, then you have this VDJ sequence. In your gene segment, in your chromosome, you still have all of those constant regions. All right, so why do you make IgM at first? Because when you make your mRNA and you start your protein translation, the protein synthesis is going to read through the VDJ. It's going to read that mu constant region right here. Your mu constant region right here. Right after that constant region, there's a stop codon. So protein translation will terminate. So that's why, even though you've got 6 or 7 constant regions still there, that's why you're always going to make just that IgM. All right? Then, once you get whatever signals to go through your isotype switching, you have that splicing of the mRNA that cuts out whatever the middle is, puts it to whatever the constant region, in this case, the constant epsilon region. So when you then have protein translation, your VDJ is going to be paired with your constant epsilon, and so you make IgE. Does that help a little bit? If not, you'll hear all of this again from Dr. Prater in a couple weeks. And hopefully she can find some combination of words that I haven't gotten to yet. All right. So, T cells mature in the thymus. They reach maturity in the thymus. They leave the thymus to the periphery if they've passed those 2 checkpoints, and then they become activated. So remember, mature but naive. B cells leave the bone marrow in an immature state, in that transitional B cell, and then they finish their B cell maturation in the secondary organ, in the spleen, in the lymph node. And they kind of like it there, so they tend to stay there. Not a lot of B cells are found in the periphery as a general rule. All right? And here is our summary. So that just kind of pulls all the pieces of these processes together and puts them in maybe some new images. For very complex processes like this, I like to try and include different images at different points in the lecture, so hopefully you will have one that you get the right visual cues that it clicks. Yes, ma'am?
The isotype switching has the constant region changes for light and heavy.
Only for the heavy chain. So the question was, in isotype switching, the constant region changes for the light or the heavy chain? No. The constant region, so we'll have, yeah, let me go back. Okay, so here's our antibody structure. So you do have a constant region in the light chain up there in the Carolina blue, right? So that is basically just, it's constant because there's enough conserved sequence that it can bind to the heavy chain, all right? It doesn't have really any function beyond that. So it doesn't matter that it changes. So the constant region for the antibody light chain, for all of the T cell receptor chains, there's only one, okay? And it is the same for all of those. I mean, same within each of the individuals. Did I lose you there and say that? Okay. But all V beta T cell receptors, all beta chain, all alpha chain, each have their own constant region, but only one. Where there is a difference is in the heavy chain of the antibody, of the B cell receptor, because that constant region is what then binds different molecules and gives different effector functions to your different antibodies. So there are different FC, and this is just to try and help make this clear. This is not anything I would test you on, the next couple sentences I say. There are different, at least on this upcoming exam, there are different FC receptors. Some of them only bind IgG. Some of them only bind IgE. Some of the isotypes bind complement a lot better than others. So there is a differentiation of function, a specialization of function. IgA, that gets across our mucosal barriers really well. So that's why we have a lot of IgA, not in the blood, but on our mucosal surfaces. Right? It's that specialization of function, which is all mediated at this level through those heavy chains on the isotype of that antibody molecule. Okay. So that's why when you go through that isotype switching, it's only the heavy chain. Does that help make things any more clear? All right, I'm not seeing any nodding heads, but I'm not seeing any tears either. I'll take that today. All right, so, yeah, we covered all of this. All right. Summary. All right, and now here's just a comparison so that you can understand, because a lot of what we've talked about for the past 2 hours, excuse me, the same thing happens for B cells and T cells, but there are some differences, like the isotype switching, like the ability of B cells to go back and make multiple light chains. So this table just kind of summarizes that comparison and contrast. Compare and contrast those two processes so you can see where there's overlap and where there's differences. All right, and then just remember, ontogeny. You've got both contact dependent, so you've got to see that MHC, you be the T cell, that MHC peptide. If you're a B cell, you've got to be able to interact with those stromal cells to get those signals. But then there's also the contact independent factors. Lots of cytokines that are being made. Flashback to all of those colony stimulating factors. IL-7, IL-2, those growth factors that occur. All of the cytokines that are going to come into play for the isotype switching that Dr. Crater will talk to you about. Then, remember these receptors are very specific. Highly unique sequences that are expressed. One sequence, one receptor per cell. So we've got that clonality. We can make our army of clones to respond to whatever the infection is. And then this process has to be tightly regulated. We don't want to go through all of this process and then have a bunch of B cells or T cells that are specific for self, that will then go out and give us rheumatoid arthritis or diabetes or, whatever autoimmune disease you like to think about or you tend to think about. Right? And that's the checkpoint. That's those processes of positive and negative selection. All right. Question. Is it as clear as mud? All right. Yes, ma'am. Yes. To a biochemist, no. So affinity is, the way affinity is a single cell affinity, avidity is the accumulation. So when you're thinking about, that a single cell is going to have multiple receptors, avidity is sort of the sum total. Jesse, another hand? So I thought out of my periphery. Okay. All right. Congratulations, you've made it through